The red one is microRNA and the below one is messenger RNA. So this is typical structure of messenger RNA: 5' cap, 5' UTR, ORF, the protein coding part, and the 3' UTR. The microRNA comes and binds the complement region in the 3' UTR of a particular messenger RNA. If both microRNA and messenger RNA are there in the cytoplasm, this microRNA will goes and binds to the 3' UTR and it blocks the translation. Within this microRNA, this is the seed region. There is 2 to 8 nucleotide regions, or we referred as seed region of a microRNA. This is important in the function of microRNA. If the complementarity is high, then the messenger RNA is degraded. But if the complementarity is less, translation is getting suppressed. Terminology here I want you to remember is this is authenticate target. Okay, so this is the microRNA and it goes and binds and blocks the translation of this particular messenger RNA. So this is what scientists known while they found the microRNA and how the microRNA regulates the protein expression. After that, research reveals that the microRNA can goes and binds anywhere in the messenger RNA and blocks the translation. After that, there are so many discoveries have been done related to microRNA and how the microRNA works. We have to think about this. The first one is abundance of microRNA. There are number of microRNAs present in a cell. The copy number also varies. And the stages of infection. This is important in viral infection. microRNA regulates the viral protein expression. So the presence and absence of microRNA in a particular cell can initiate or suppress the infectious cycle of a particular virus. Rate of repression of expression. So this is the speed, how fast a microRNA can stops or blocks the translation of a particular messenger RNA, or how slow. So seed match, most microRNA can go and bind where that region is called seed matching and the rest of them are called non-canonical matching. So the difference between these two: transcriptional and post-transcriptional targeting. Some microRNA can go and bind while the transcriptional state. Most of the microRNA can act as the post-transcriptional targeting agents. So this one is very important. A particular microRNA can have many targets. So it can go and bind to the different types of messenger RNA and blocks many number of protein synthesis. And the converse is also true. Many different types of microRNA can have a single messenger RNA target. Direct and indirect repression. There's a possibility of indirect repression of a protein also reported, as we will discuss in this presentation later on. 3' UTR length can change during different stages of development. This is also making some importance. This is the reason of temporal and spatial difference of protein expression levels in a cell, and also splice variants. Mimics versus original. During last decade, some scientists identified that they created an artificial microRNA in such a way that it is complementary to the particular microRNA, and they introduced into this cell and blocks the particular microRNA and its effects. So they called those microRNA mimics. So but later on, the scientific community identified that within the cell, some endogenous non-coding RNAs are there that acts as a mimics for some particular microRNA. We are going to discuss about those in later. Synthetic transcripts containing mRES, a competition with target mRNA. What is mRES? This is microRNA responsive elements. Okay, M for microRNA, R for responsive, and E for element. So what is actually the M or ES means? In a particular RNA, whether it is coding or non-coding, if it is having binding site for any microRNA present in the cell, we refer that region as microRNA responsive element. So it may be present in the mRNA, or it may be present in the other non-coding RNA, or it may be present some circular RNAs also. So we refer these sites as M or ES. In a particular experiments, one person, he synthetically designed a plasmid and he put a particular transcript which is having multiple M or ES for a particular microRNA. So he introduced this plasmid into a particular cell. Within this cell, this transcript codes for RNA. That RNA contains multiple microRNA responsive elements, that is binding sites. So all the microRNA present in this cell, which is complementary to this M or ES, binds with that. The synthetic transcript containing M or ES nullify the effects of that particular microRNA in a cell. So he hypothesized that in a cell there is the possibility of some such RNA can have some M or ES for particular microRNA, so it can block the microRNA responses or functions in a particular cell. These experiments leads to the discovery of competitively endogenous RNA. So before we are going to appreciate the experiments, we have to stick with the terminologies, because in this field there are many number of terms that is confusing here and there. So first, the coding RNA and non-coding RNA. So here I want to mention you that some messenger RNA can also act as non-coding RNA. miRNA target I emphasized in the previous videos, so refer those. And microRNA responsive elements we have seen just now. Competitively endogenous RNA. So why we named competitively endogenous RNA? So which is endogenous, which means it's synthesized within the cell, and competitive refers that it is having multiple microRNA site, so it competes for microRNAs. And circular RNA, as we will see in this presentation, some RNAs are there in circular form. It also can act as competitively endogenous RNA. What's the character for competitively endogenous RNA? So it should compete for at least one microRNA. Okay, that means it should have at least one microRNA responsive element in its sequence. So what are all the RNA which is having M or ES? We can consider those RNAs as competitively endogenous RNA. So there is long non-coding RNA also there in the cell. If it is having M or ES, that is competitively endogenous RNA. And target mimicry. We referred the target, the microRNA target is situated in a particular messenger RNA, and while the microRNA goes and binds, it stops or it represses the translation. So target mimicry is nothing but different types of RNA which is having M or ES. We can express this in the term by using target mimicry. And microRNA sponge. This is another fancy term in literature. We refer these competitively endogenous RNA as microRNA sponge. Why? If the competitively endogenous RNA is there in the cell, all the microRNA goes and binds. Okay, it sequestering the microRNAs and it nullifies the microRNA functions. So we refer these competitively endogenous RNAs as microRNA sponge. In this slide, I animated some of the main events that occurs in the cell. So there's a nucleus, and within the nucleus, DNA is there. The RNA is transcribed from the DNA. So this is messenger RNA. As it is having introns, these introns are spliced out. So there's a possibility of these introns can circularize and it can form exonic circular RNAs. Remember, these exonic circular RNA may also have M or ES, so that we can refer these exonic circular RNA as competitive endogenous RNA. And after that, mature microRNA is produced. Note that the matured microRNA is shorter than the previous. So these two RNAs came out to the cytoplasm and ready for its function. So in the same cell, there's a possibility of synthesizing microRNA. In some other places in the genome, some hairpin precursors are there, so it transcribed and some DROSA and ricer enzymes are cleaved out and it produces matured microRNA. So these mature microRNA come out from the nucleus into the cytoplasm and it's ready for its work. So in a particular nucleus, somewhere else in the genome, there's a possibility of coding non-coding RNAs. So these non-coding RNAs also synthesized within the cell nucleus and it also comes out for its functions. So cytoplasm contains all these different types of RNAs. First, this messenger RNA can start protein coding. This blue one as the ribosome, so it can goes and binds to the 5' cap of a particular messenger RNA and it can start synthesis a particular protein, and this protein is having some functions in the cell. This is the first case. So in the next case, the mRNA is also can start the protein synthesis, but at the same time, if the cell is having microRNA and this microRNA is having target on that particular messenger RNA and it blocks the protein synthesis. So what about if there's a long non-coding RNA in a cell and also circular exonic RNAs? If these circular exonic RNAs or long non-coding RNAs or whatever else the RNAs which is having the competitive site, binding site for microRNA, all the microRNAs are going and binds to the ceRNAs. So this particular messenger RNA participate in translation and can produce protein. So remember, due to this effect, we call these ceRNAs as microRNA sponges. So we refer this phenomena as competing endogenous RNA hypothesis. So this hypothesis having two parts. In some paper, they may refer that these non-coding as well as coding RNAs are talking to each other by the microRNA binding responsive elements. So this messenger RNA and this circular non-coding RNA or linear non-coding RNA, whatever it is, these are cross-talked or these are communicated by the presence of microRNA responsive elements in the particular RNAs.